A special welcome to TUT students who are our primary audience today. And today's speaker is very special. I will tell you my story before I introduce him. So when I was a student, as I said in our previous talk, that public health was my favorite course. I loved going on internet, reading things. And then one day I got into his paper, one of his papers. I read, then it became a habit. I would go and read one of his papers. And this other day when we were going for meat inspection, I don't know how I got his PowerPoint on meat inspection, and I read it beforehand. And when I went, I was shining in the class. I was answering questions, and he became like my role model. And when Karen asked us to have this series for the TUT student, I thought of him, and I went straight on internet, got his email, sent him an email, and I'm so happy that he agreed to be our speaker today. And this man, he's not just a man, he's a professor. He has worked for more than 20 years in One Health, dealing with 20 years dealing with zoonotic diseases, and he's a 3-in-1. When I say 3-in-1, he's a lecturer at the University of Zambia, Unza, and he's also a veterinarian, and he's also a lecturer for public health in the University of Unza. He's also the coordinator for One Health, working with COHESA. So would you please, ladies and gentlemen, help me welcome Professor Musso Munyeme, who is our speaker today. Professor, welcome, please.
Thank you very much, Sarah. Thank you, Karen and Mareth. Welcome all to today's presentation. Like Sarah has introduced, I'm not going to introduce myself. I've really done a lot of work, and it's good that one of my components has been meat inspection, not only for domestic, but in wildlife. Yeah, I've done a lot of meat inspection across different animal species, both domestic and the wildlife. So to start today's lecture, you excuse me, I will just remove the video so that we concentrate on the slides. The topic which I'm going to start with today is based on our own studies which we have done in Zambia and a bit with our colleagues outside Zambia, especially the neighboring countries. So we are looking at the spatial temporal distribution and also the genetic diversity of Bacillus anthracis. This is the bacteria which causes anthrax. And in Zambia, we say this has been modulated mostly by the environment, the anthropogenic, those are human activities, and wildlife factors. And because of these, the environment, human, wildlife factors, we are talking of a One Health issue. And like Sarah has introduced me, I'm a country multiplier for capacitating One Health in Eastern and Southern Africa, based in Zambia. And that's my name. So let us look at anthrax. One thing we should know about anthrax is that anthrax is a classical zoonotic disease. So what do we mean by classical zoonotic disease? You see, there are diseases which are zoonotic, but they will depend maybe on someone getting the disease through a certain media. But with classical zoonotic diseases, you are going to be affected directly. For anthrax, it means that infection is directly from the source, directly from animal to humans. So anthrax has emerged as a very serious ecosystem problem. It's a very serious ecosystem issue. It is affecting humans, it is affecting livestock, it is affecting wildlife, and mostly we have seen mortalities. In Zambia, if you look, starting 2022, 2023, last year, those who might have read, we had the serious outbreaks of anthrax, not only in the wildlife, in hippos. They affected the cattle. But what shocked us was the human outbreaks. There was a rapid spread in human outbreaks. But in Zambia specifically, we've also seen the devastating effects on livestock production, on tourism, as well as on general public health. And that is why it makes it a classical One Health issue. When we look at the preliminary trend analysis in Zambia, and I think even in some neighboring countries, I know the issues in Tanzania is the same. We have been having what we call sporadic annual outbreaks. Mostly in Zambia, it's in the upper Zambezi region, especially the human and the cattle anthrax. But if you go to the eastern part of Zambia, very close to Malawi, we've got what we call the Luangwa Basin. There's the Luangwa River there. That's where we have been having sporadic outbreaks, specifically in wildlife, the hippo population. For those who like reading, like Sarah, if you read 1988, we had a serious outbreak of anthrax. We lost almost 2,000 hippos to anthrax in the Luangwa Basin. In 1997 to 1999, we had almost a similar situation. Surprisingly, around 2007, again, we had a similar situation of losing hippos in the South Luangwa. And the West again came in 2011 in the South Luangwa ecological basin. One aspect about anthrax, others call it a soil-borne disease, where there is a soil exposure, because if you look critically, because of its way of sporulating, there is a soil exposure being a function of the grass biomass, but also a variety of ecological and anthropological factors have to be considered when you want to determine what we usually call the causal mechanism. I know most of your students, I think you have read about the theory of causation. So feel free to read on the theory of causation. Once you've read on the theory of causation, and you come and you look at the way anthrax infects, especially herbivores, because these are the bulky grazers. The most important herbivore, which is a bulky grazer, is a hippo, because the hippo, it even picks the spores very low. It pulls the grass, pulling together with the roots. And by doing so, it's even pulling the spores. And this brings me to what we call the common form of anthrax. If we look at the common form of anthrax, one of the most important aspects is that this is a disease which is peracute to acute, and it is a multi-species zoonotic disease. It's a classical zoonotic disease, but also multi-species. There are zoonotic diseases which are specific for a certain specific species of animals. But anthrax, it will affect a lot of different species, apart from also affecting human beings. When we are talking of peracute, it means that the disease occurs almost without premonitory signs. In a livestock, you find a herder wakes up in the morning. As he's trying to head the animals out of the crow, all of them look very okay. Maybe they start grazing. As they are grazing, just sees an animal collapsing, dying. The next thing is seeing blood is oozing from all the natural orifices, from the nostrils, from the mouth, from the ears. Sometimes I've seen even the blood coming around the eyes because of the nature of anthrax. No sign that the animal was sick, just collapses. And that's what we call peracute. So what we see is that this bacteria usually is caused by a spore-forming bacteria, Bacillus anthracis species, because there is also Bacillus cereus, but these are specifically under Bacillus anthracis. And it in the host, it remains in what we call the vegetative form. But when an animal dies and the anthrax is exposed to oxygen, it forms a spore, and we call that sporulation or a cyst. So when there is enough oxygen, the vegetative form immediately changes and sporulates, which forms a very hard, difficult to deal with spore, which can actually remain in the soil for at least 60 years. So that is 6 decades in the soil. That's how anthrax becomes very, very dangerous. Now, its pathogenicity is owed to 2 virulent factors, because there is a virulence and there is also the other factor which causes it to move a bit faster. So when you are looking at the virulence, there is the formation of a poly-D-glutamyl capsule, which mediates the invasive stage of the infection. Then once it is in the body, there are toxins which are produced. These are production of multi-component potent toxins, anthrax toxins. These toxins, we have got 3 important toxins. There is the protective antigen, the lethal factor, and the edema factor. So when we move a bit, let me move a bit. I want to show you this slide. When the gram-positive spore-forming bacteria, I want you as students to look at this. When this is the vegetative form on the left side, the vegetative cells. But the vegetative cells, once they are exposed to oxygen, you can see on the right side, the spores. You can see the spores like in blue stain, methylene blue stain. So once there is carbon dioxide and the nutrients, then in the body, the spores now go back to the vegetative form. But the spores, like I've said, can remain in the soil for over 60 years. And anthrax has also been categorized as a category A bioterrorism agent. And this is a very important aspect. Now, I brought some of these pictures from our own field studies. I want to show you, like we have outlined, that the anthrax is a multi-host zoonotic disease. Now, the Bacillus anthracis in humans, let us start with on the top left. You can see a human there. I talked about the edema factor. There is also the lethal factor. Now you can see the effect of the edema factor. Someone is all swollen up, but there is also the sore, the eschar, which forms. This is the cutaneous form of anthrax, which is affecting this person here. There is also the form which affects both lions and dogs. If you see the lion, the edema aspect, it has all been swollen up. One of the aspects which we see mostly in these carnivores is that the edema factor becomes more pronounced, but the lethal factor is more deadly in herbivores. You can see down the way the cattle dies, you can see the hippo, and you find that if you look at this animal, the cow here, is frothing with blood from the nostrils. Sometimes you find even from the anal region, there is blood coming out. It's the same with the hippos. And one thing we've seen in hippos is marked bloating. The hippos become so bloated and sometimes they even burst. It's the same with elephants. These we've seen in all the study and the response we've done because of anthrax. If we go to the science, like I've said, I will need to say there are 3 forms of anthrax, especially for humans. The cutaneous form, then there is the intestinal form, and the pulmonary form. I will just, there's a fourth form, but these are the ones which are critical and well documented, well researched, the cutaneous, intestinal, and pulmonary form. So these three forms, the most common, almost, they say close to 80% are the cutaneous form, the one which forms in humans, that is the cutaneous one. But in wild animals and also in domestic animals, especially herbivores, what you are going to see is actually the intestinal and what also we say the respiratory or the systemic form. Because here, animals die without premonitory signs. The rigor mortis is usually incomplete, and you are going to see blood oozing out from all the natural orifices with severe bloating. If you look in the middle, there is a picture of a hippo completely distended because of severe bloating. And sometimes if you go there, sometimes you find there's a voucher trying to perforate, you find the whole hippo just bursting. In cattle, the top one, you can see blood oozing without clotting. This blood will continue just oozing from the animal. And this is one of the worst aspects because as it is oozing, the Bacillus anthracis is exposed to the oxygen and we end up with sporulation. In lions and other carnivores, one of the most important aspects we've seen is, like you've put in the middle, carnivores, subacute edematous swelling of the face and the neck. This is one of the most common things you are going to see when you are dealing with anthrax in animals. Now, like we have been told by Sarah, there's one issue about One Health, anthrax in nature and its transmission. When we are looking at One Health, we look at the environment because the environment is the common denominator. The environment being the common denominator, you are going to have animals because we said this bacteria, if you look at the Ganzo et al. Diagram here, you have below the ground, when an animal dies, let's, because this is wildlife, I just used a zebra to depict. You are going to have a zebra here. Then you are going to have inoculation of the soil. So what usually happens is that oozing of blood forms what we call spores. As those spores, they will go and fill the ground. But what will happens is that as the grass is growing, they will pick the spores mostly around the roots. Then during the dry period, and in Zambia, this is one thing we have noticed. Anthrax occurs when the grass, what we call the rangelands, have become maybe slightly depleted. There is a bit of overgrazing. Now, there are certain animal species like the hippo. The hippo, the way it does is that it's going to pull the whole remaining grass with its roots. Now, as it is doing that, it is also pulling the anthrax spores. And these anthrax spores, they end up in the animal. And that's where you find that a lot of mortalities usually start with hippos. Then they can go to carnivores, human, or also they get affected maybe through inhalation or cutaneous consumption. All those aspects are linked to the environmental aspect of anthrax occurring. So this aspect shows the One Health from humans, the environment, and domestic and wildlife. So this is a very important diagram. So the overarching thrust for us in Zambia, which we wanted to use so that we build this information, we wanted to know the precise pattern and the spatial distribution of anthrax outbreaks. Then we wanted to also determine the strains because up to now, the strains of anthrax and the spatial distribution had remained relatively unknown. We just see what we call sporadic outbreaks. The lack of organized zoonotic disease investigations at ecosystems level and also the prevailing anthropological activities, we have noted that these have been largely contributing factors, especially when we look at the deterministic and risk factors which are at play in Zambia. It's not only in Zambia, but also neighboring countries. I want to bring to the attention, you look at this map, this was for 2023. If you look at this map for 2023, there is what I can call the animal cases. If you look, this was 2023, the blue one on the right. The first cases, there is a, if you look, you go down, there's what we call Sinazongwe area here. The outbreaks started in Sinazongwe in 2023. And if you look at animal cases, the animal cases, deep blue, they were between 131 to 251, because the total number of cases by that time was about 552 cases in animals. That's where the outbreak was quite intense. Then there are these in Kazungula and up there in north. This is the, if you look on the north side, that is the Luangwa Basin. Here on the southern, going to the western part, is the Zambezi, what we call the upper Zambezi Basin. Now, one aspect is that anthrax was breaking out in these regions. You look on the left side, 2023, that is October to November, human cases. We had a total of 709 human cases in 2023. And there is what is known also as the lag effect. Why the lag effect? What happened is that in July, August of 2023, we had outbreaks in cattle and hippos. There were no human outbreaks. However, came September, end of September, October, November, up to December, the animal cases by September we had almost no records of animal cases. But human cases from September, October, November, all the way into early December, we had human cases. And this is because of the lag effect. This lag effect was because of anthropogenic aspects. What happened is that from the blue areas here, people started taking dried game meat into different areas. Lusaka, the capital city, got affected. If you look at the red areas where there were lots, this was because of anthropogenic from the wildlife and livestock keeping areas into commercial and what we call urban settlements, where they don't keep cattle and anthrax occurred. And this is a very important aspect when it comes to public health and the ramifications of disease outbreaks. So, when we look at this, one of our specific thrusts in Zambia in terms of looking at anthrax, we know that anthrax, if you look, there is what we call protracted dormancy. Like I said, it can be 60 years in the environment. It can stay there, making the Bacillus anthracis monomorphic, which is due to slow evolution. Thus, ascertaining the source and the spread of the disease is essential to determine the evolutionary relationships amongst the isolates. Also, one thing we wanted was to trace the spatial distribution and transmission paths. Like I have said, if you look on the map on the right side, there are two important regions, big ones, although there's a third one. There is the Zambezi region and the Luangwa Valley. The Longwa Valley is very close to the eastern part, close to Malawi, whilst the upper Zambezi is close to Angola, where we border Zambia and Angola. Now, in this map, you will see what we call historical outbreaks. The historical outbreaks are in these areas. Even now, where there is pink, that's where there's been a lot of historical. Now, in Rwangwa, this is wildlife. If you see there is a yellow star, that has been wildlife anthrax. Then, in Western Province is a red star. It has been cattle anthrax. Now, one important aspect is that in both the Rwangwa and the Western Province outbreaks, they've all ended up in human beings, hence the zoonotic nature. So, the other objective, as we are doing these studies, was to establish, and we are still doing a lot of studies around this, the genetic diversity of this Bacillus anthracis, and also to determine factors which influence the distribution. In terms of laboratory undertaking, we've had a lot of studies. One of the studies was that we had to undertake ethical approval from the National Ethical Board. And what we had to do, we had to face what we call field studies, where we used to go out in the field to collect biological samples from wildlife, from the environment, from even carcass remains. From humans, we are getting from wound swabs. And fortunately, we were allowed to use the biosafety level 3 at the University of Zambia with the School of Veterinary Medicine. So this is just the flow diagram of the way we were doing the determination, confirmatory determination of anthrax. From blood cultures through DNA extraction until we do the whole genome sequencing, and then we do the bioinformatics, reference mapping, synapse detection, the single nucleotide polymorphisms, and the index detection. So, from culture, we were always very cautious. We were working, like I've outlined, under biosafety level 3, where the samples were cultured on blood agar plates. They were examined for the typical colonies after an overnight incubation. Suspected colonies which we are picking were those from great white between 0.3 to 0.5 centimeters in diameter. And most we are checking the non-hemolytic with ground glass surface appearance. If you check the colony characteristics, with sometimes, if you look, you will see the medusa head or held hair appearance, which is in this slide. We also did microscopical examination because we didn't want to miss anything. So all the kind of biological as well as laboratory procedures had to be undertaken. Microscopic examination from all the areas. We looked at the gram-positive large rods chains, sharp edges, and those in the endospores, especially when we are looking at the McFadden stain. We also did molecular work. One of the things which we had to do was to run what we call a multiplex PCR, which, like I said earlier on, there were other factors like protective factor, which we had to look. Then we had to look at 16S sequence for identifying the universal primer for identifying that we are dealing with a bacteria. Then we had other genes which we had to look for, the CAP, the BA. These identified that we are dealing with anthrax. Once these were taken into consideration, we were able now to be very satisfied that what we are dealing with was anthrax. And we went from these into whole genome sequencing. Now, I want to show you, if you look at, forget about the sample ID, but I want people to look at the location and the source. So if you look at the location, Lower Zambezi, I think you saw the map, Lower Zambezi borders Zambia and Zimbabwe as well as Mozambique. It's near Zambia, Mozambique, and Zimbabwe. Here we were getting samples as early as 2011. These were from elephants and from hippos, as well as from humans. I like this one. This was a 2011 and 2013 sample, Chama human, which is H29, Luangwa Valley. Then we had also Luangwa Valley samples. We had also, if you look on the left side, samples coming from other animals like impala, then Western Province from cattle, Western Province from humans, and also from the environment. If you check also in all these, we are also getting environmental samples. So sources were from the environment, from humans, from wildlife, and from domestic animals. And this was a proper One Health issue. And these samples, we had to subject them to whole genome sequence. Now, the phylogenetic relationship, which determines the closeness as well as the relationship, or to show the tracking, how closely related are these strains. Now, we did from a period of 2013 to 2017, then we moved again to a period from 2013 to 2020. We did also bacitracin susceptibility tests in anthrax. These are different types of tests which we are doing. Now, one important isolate is the human isolate, which was found from Shangomo. We put it as a SHH20. These are some of the anthrax strains which had the large genomic deletions, especially those collected around 2017. So what we discovered is that there are also some human isolates from the Western Province which were found very far away from the area, almost about 1,000 kilometers apart, which showed that there was a bit of anthropological aspect in terms of spread of these anthrax infected meat. But we also went further to look at the Zambian isolates with other global strains. Now, there's something which was very, very, which we looked at, and it came out a bit unique. So what we saw is that the anthrax strains in Zambia, you remember I showed you the map where there was a western part which is near Angola. We had some strains near Angola on the Zambezi areas, which are more related to the Southern African strains, the strains which are found in South Africa, like SA. If you look on the diagram, there is SA 020, which is a South African strain. Then there is also AF03, which is from South Africa. This was in the database. When we blasted and did the comparison, we discovered that most of these strains, apart from being closely related to the South African, the Zambian strains were also closely related to the Tanzanian strain, especially the wildlife and environmental isolates in the northern eastern part of Zambia, closely to Malawi, what we call the Luangwa Valley. These are closely related to the Tanzanian isolates, which were isolated from the Ngolongolo isolates. So when we looked at most of these, we discovered that most of these isolates had also a relationship in wildlife. Now, the question was, what is the role of wildlife in this? So is wildlife involved in dispersal? Our current study, as we are talking now, we've got some PhD students who are trying, that's why I've put it in red, who are trying to unlock this question, because as we are doing most of this research and generating new knowledge on anthrax, we are trying also to see how we can bring in new knowledge and understand these aspects. And one of the students is trying to do what is known as ecological niche modeling based on the genetic information and the distribution of these anthrax. Now, based on this, there is what we call the take-home message. What we've discovered is that with the Zambian scenario, the western province of Zambia has the most genetically diversified kind of anthrax, which is influenced by annual outbreaks in livestock and humans due to what we might term a shorter dormancy period, because here we've got almost annual or yearly outbreaks. Then two strains, like I've outlined, the SH20 and the Chama H29, these are dispersed human isolates between Western province and eastern part near the Luangwa, near from the Angola to the Malawian side. So these are key, and we want to see further what the anthropogenic factors are at play. There is a student, Kamboy et al., there is a study et al., they are working on this and trying to bring more information. So the Luangwa Valley strains have been found to be closely related to the East African strains, while the lower Zambezi and Western province more to South African strains. So one of the angle in which we are trying to move is to understand the relationships linked to wildlife conservation corridors, because I think you have heard of transfrontier conservation areas in relation to wildlife conservation, but we want to follow these and understand how these are affecting or abating or playing a role in anthrax dissemination. One of the recommendations is that there is need for strategic livestock vaccinations to be routinely implemented, especially in the western province of Zambia, where we've been seeing a lot of anthrax in the domestic aspects. We need also bottom-up One Health-centered promotion in terms of education, awareness, especially amongst the community, because you find that when there are outbreaks, animals die on themselves, and the community, they go and start consuming, and they end up with all sorts of these outbreaks in humans. So the strengthening of One Health, both at a coordination level, collaboration, implementation, and also making sure that these task forces are maintained even after the public health emergencies have gone down, are kept as a policy. We want to ensure that there is an improvement in preparedness and utilization of what we call common pool resources in terms of sharing of grazing land, sharing of drinking water points for humans, for animals, livestock, as well as wildlife. Those aspects need to be taken into consideration. Once we do that, we'll be able to understand and be able to respond to the issue of this classical zoonosis anthrax. So basically, this is what I wanted us to share for today. And I gave the presentation to my colleague Sarah to finalize. So Sarah, maybe we can now entertain questions from the students.
Thank you so much, Prof. This was so insightful. And there's a lot that I've learned. But then before the student, I know they're getting ready with their questions. Genesis said she has a lot of questions. But then I have one question for
Please.
you.
Go
How
ahead.
were you disposing the carcasses that have died due to anthrax?
That's a very important question. That question came up all the time. One of the key aspects is, and I need to inform the students here, there is what is known as quicklime, caustic soda, because caustic soda will even eat the bones and even the spores. This is the only chemical we've seen. But I was shocked at one time to find some of the colleagues who didn't understand. They were just using ordinary lime. someone told them just use lime to bury. So what we are doing is that we are digging a pit. We would do even burn when we had the fuel, but most of the time where we were failing it to burn, we were using caustic soda. This is quicklime. That is what we were using most of the time. If we didn't have the caustic soda, we were burning. We use fuel, we burn as much as possible and bury. So that was one of the things, because when we identify the area, we are getting geographical positioning system, the GPS, and come back sometimes just to decontaminate the place again, where we do mix the soil with caustic soda, just to make sure that at least we've done something which will prevent the animals, because one thing I will tell you is that we did another study where you put like a mark, let's say it's in the center. After about 3 years, we went around, we made what we call transects around that area. We were able to pick spores 30 meters, especially where there is a sandy soil, 30 meters from the point where we had buried if there was no decontamination. And that was dangerous. So that is one of the aspects which we have discovered with this work with anthrax. The spores are real. 3 years after burial, and there's been no proper decontamination, we are able to pick the spores away, especially on the windward side of the, because there's a way wind moves mostly in a prevailing direction. We are able to pick those spores. Yeah. So the key is to decontaminate or to burn the carcasses. And that's what we are doing in disposing. Yes, thank you.
Thank you so much, Prof.
Sela, I think Letitia has a question in the chat.
Okay.
Thank you, Sela, for opening the mic. I just wanted to ask, doctor, I think it's doctor, right? Do you use the strain?
Yes.
Yeah. Do you, in the lab, do you use the strains with the attenuated virulence?
Are you able to come back again? You talked about, do you use the strains with the attenuated?
Yeah, virulence.
Oh, yes, attenuated virulence. Yes, one thing I need to say is that, yes, we use the strains also for control with attenuated virulence. We've got our colleagues from on the Depot. We work a lot with Aisha from South Africa. She actually gives us a lot of support when it comes to giving us controls. Yes. So Aisha, she's part of our team with the there's a lady there, Edinburgh. I've forgotten the name, the supervisor to Aisha. Yeah, so we work as a team. Yes. Maybe Kellen might know the lady in South Africa.
I've heard of Aisha, but I can't think of a supervisor's name at the moment.
Yeah, Aisha, she's now self-supervising. She has gone beyond the. She's a mentor now. Yeah, she's very good. So she provides us with those. Yes. So with Letitia's question, yes, we use those attenuated strains a lot actually. Yeah.
Thank you.
Thank you.
That's good to know. Yeah, and my other question is, during your talk, you talked about the vegetative form, which I think is the virulent one, the pathogenic one,
Yes.
and then the sporadic one, I think.
Yes.
And then when you grow it on your agar plate, which form do you get?
When you draw it with the other?
On the blood, on the agar plate.
Oh, yes.
Yes.
That's a very good question. That's a good question. You see, on the blood, what we want is even when you go in the field, we've gotten even the spore form itself, we've gotten from the environment. So there is a way we do the what we call pump priming. Sometimes in some way to hit some of the samples to make sure that we stimulate the spores. Once they are cultured now on the nutrients, you will find that the spores now are able to do what? To grow. Most of the time, very less likely that you are going to deal with the vegetative form. Almost all the time, actually, you'll be dealing with the spore form. Yes.
Thank
So
you
it's
very much.
mostly the spore form. You are welcome. Oh. Mhm.
Thank you so much, Dr. Letitia, for the good questions. And now I can see Lucy has been raising her hand for so long. Lucy, can you unmute and go for it?
Everyone.
Evening, Lucy.
Evening.
I'm a TUT student. I'm concerned about this thing about hippos when they die in water. Professor, hence these hippos have been infected with anthrax. What do you do to purify or clean that water? Because let's say I'm a student doing my will on a certain
Mhm.
reserve, and
Mhm.
we find that there's a hippo that was infected with anthrax, and it has to be
Mhm.
taken out of the water. Isn't that
Mhm.
water infected with anthrax?
Yes, good question, Lucy. So I think Lucy has got a number of questions. One of the questions she has asked, I think, is linked to human patients. What happens when people have anthrax? So I need to inform you, I've been in the field and I've noticed quite a lot of scenarios. One good thing, like if you remember when I was giving an explanation, I said the good thing is that over 80% of the cases we see are usually cutaneous form. But in North Luangwa, with my colleagues, we saw it was quite bad, the GIT form for the human. And unfortunately, that patient didn't survive. And like you have outlined, there was a lot of decontamination, and the hospital people had to decontaminate the ward, the beddings. It's almost the way they do it in these like Ebola situation, because these are quite bad zoonotic diseases. And because of that, it's an occupational hazard, even to the people who are working in those hospitals. Anthrax, it looks simple when it's cutaneous, but the gastrointestinal form, even the respiratory form are really, really bad. And the hospitals, they've got biosafety procedures which get activated once a case like that has occurred, and they understand. They follow the standard operating procedures. They've got soaps on how to decontaminate. Sometimes they even fumigate what we call the isolation wards, because these have to be very, very separate. So they've got proper procedures to handle such scenarios. Yeah. Now, coming to your last, the other question you talked about, are you able to repeat again, Lucy? There was a very good, apart from there too, unless if I've missed
My
the second part.
my other
Uh-huh.
question was, doesn't that hippo contaminate the water?
Yes, the hippo, yes. Mhm.
And
That's true.
what do we do as nature conservationists to prevent that water from affecting
See,
other animals and people?
that's a very good question. And there is what is known also, this, it's bimodal. There's what is known as the dilution effect of the water. Most of the animals which die, but there is a danger in that, because not all the spores which are in water just get diluted and inactivated. That is why we've seen that there are some spores which have been carried from the northern part, same spores in the Ruangwa, downstream. We've seen outbreaks happening 130 kilometers upstream, going downstream because of that aspect. So it's very difficult to deal with such scenarios. That is one aspect, because how are you going to decontaminate a water body? The water is ever flowing. So this is where sometimes we just leave it to nature, because one, it is almost impossible to decontaminate these flowing rivers. What chemical can we use? Because there are also biologists, there are also nature conservationists who say we cannot put any chemical in the water. You remember, in these water bodies, it's not only the hippos, there is fish there. There are people who are using the same water down the stream somewhere who are drinking that same water. You cannot kill one species to protect another species. So what usually happens is that the natural ecological systems, what we call health services, which the ecological aspects occur, ecosystem services, they will just deal with themselves. And one of the ecosystem services of water is what is known as the dilution effect. It does work, but it needs further elaboration. More work needs to be done, maybe on a small scale, to understand the dilution effect of water in terms of controlling anthrax. Thank you, Lucy. I like that question. Thank you.
Thank you.
Thank you so much. That was a good question. And thank you, Prof, for the good answer. And yeah, before I go to Red Tabire, I would like to go in the chats. Genesis, you have typed a question. Do you mind unmuting and asking it? Okay. As we wait for Genesis, we can start with Red Tabire.
Good evening, everyone.
Good evening.
Good evening.
Prof, I had a question about the control measures or control methods that you have been using to control or to lower the anthrax cases in humans.
In humans.
So
Yes.
which one of those control methods or measures was most effective?
Thank you. One thing about anthrax in humans is that it is as a result of anthropogenic activities, illegal meat movements, someone steals meat from dead animals. So what we usually do is that in humans, there is also what is known as a prophylactic treatment. When there's an outbreak, humans can actually be given antibiotics. Currently, one of the good antibiotics is ciprofloxacin, has been working very, very well, but it is something which is only recommended in situations of outbreaks. The normal penicillins, they do work very well in anthrax. But when it comes to control, we have to control anthrax in livestock before it jumps to humans, because in humans, it comes because as a result of a failure to handle the situation, either in the environment, failure to decontaminate those areas, or improper handling of carcasses, or failure to vaccinate livestock in what we know as the high-risk areas. So the high prevalent anthrax areas for where we know livestock is almost an annual basis, we need to start vaccinating. We are going to prevent the disease jumping from livestock to humans. In wildlife, that's where we need now also, if you look, I talked about education, awareness raising, and also community conservancy coming in to prevent this occurring, identifying quick surveillance and bringing in this what is known as village intelligence, vigilance. When an animal has died.
There he is. There he is.
Yeah. Someone wanted to call, I had to cut him. My internet is stable. Someone is trying to call.
Okay.
Yes. Yeah. Was it answered?
We lost you on the last part, but
that was Tavira's question, eh? Yes, so I was talking about preventing the disease jumping from either dead animals or from livestock into humans by putting in measures, as well as the conservation teams are putting in aspects which can prevent the crossing over, because there is what we call the wildlife human interface, where they interface. So we need to put a stoppage to that. Yeah. I think there's another question from Genesis.
Yeah. The first one was more similar to this, where she says, in regards to the environment playing a huge role in distribution of the disease, especially in terms of spores, has there been any studies to reduce the role of environment that the environment is playing, or methods such as eradicating them from the soil or plant matter the animal consume, or cleaning the water that the wildlife consume?
Yeah, that's a quite mouthful question. One aspect when we are dealing with the environment is that the environment, you see, the environment is our common denominator. Humans get water from the environment. Animals get water from the environment. We get food from the same environment. Animals get food from the same environment. Even pathogens, they exist in the environment. Anthrax is found in the environment. That is the common convergence point. So, being the common convergence point, the environment, like I've said, being the common denominator, we need to handle it with care, because the environment, there is a saying where it says that the environment does not forgive. So we need to handle the environment with care, because once we mishandle the environment, we bring in certain aspects which might harm us. The environment will surely expose us either to those products, because even the products we are using now to try to decontaminate the biologicals, there is you might not know, there is a colleague of mine, very controversial, who actually supports the existence of these pathogens in the environment. And he's saying, we are not aware that these same called pathogens which are causing the disease, they might be there for a reason. Once you wipe them out, we might create something even more deadly. So the environment has got a way of taking care of itself, but it's us humans who would want to put in measures which are not in conformity with what the environment needs. So that's where now, what is the common convergence point? We need to be very careful on that one. So usually ecologists, environmentalists, they will give a cautionary aspect on that one. And for us who are public health practitioners, we also understand and we leave it at there, because they say that sometimes changing the environment, you might just create what is known as a nutrient concentration point for these pathogens, which you might now lead to an explosion. So these are factors which we look at delicately and usually have to be cautious. Thank you.
That's very interesting. There is some truth in what we have collocated, that everything that is in the environment is there for a reason. Yeah, and also that if we complicate the control, we might end up like bringing a bigger problem. That's so true. I think we just need to keep it simple. Okay, so a second question from Genesis is, why are herbivores more affected? And why is the disease more lethal to herbivores than compared to carnivores and humans? Why? This was also my question at some point.
That's a very good question. You see, with the herbivores, one of the things we talk about is also the quantum of infection is one thing, because the amount of the amount of spores which they ingest. Then the route of exposure, because herbivores, they actually get it direct. It's what is someone said, direct injection, like in vehicles. They ingest it direct into the GIT, and then it's assimilated into the blood. So when there is that direct route of exposure, you can just imagine the lethal aspect of the disease. It's like you've just injected it direct into your system. So that's where it becomes a very, very dangerous. And then, like you have said, the susceptibility, because these animals, they are quite susceptible. Their immunological makeup, because if you look at the immunological, I don't need to go to these aspects about the immunology. The immunological makeup of herbivores is different from that of humans. And that's one, it's quite a detailed topic on its own, where you have to look at the toll-like receptors, you look at what is really happening with the immunological upregulation, downregulation mechanisms. The way they respond to these toxins is different to the way other species will be. So the species specificity and its own immunological makeup also makes them very susceptible. And the herbivores are susceptible, starting from the way they are going to get the exposure, which is almost a direct injection, which is a very, very, very direct into the GIT and into the bloodstream, making them highly susceptible than other species. But if we look at that, we have carnivores, which will ingest animals which have died, but they don't get affected the way herbivores get, because most what happens is that the carnivores, they will be eating an animal which mostly has got these lethal factors, the toxins. So there are all these different aspects apart from the immunological aspect. Yes.
Thank you so much, Prof. And thank you, Genesis, for the question. There is another very interesting question by Kagiso. Kagiso, would you please unmute and ask your question?
Evening.
Good evening.
Yeah, so Prof. Musso, I wanted to ask if ever does anthrax also affect vultures? Because I think people know that vultures are immune to many wildlife diseases.
Yes. Thank you. I had a colleague in Zimbabwe, Professor Peter Mandi. Maybe you might have heard of Professor Peter Mandi. Very good colleague of mine. Unfortunately, he passed away. We used to call him the voucher. That was our nickname for Peter Mandi. We used to call him the voucher. They got meat, which from an animal which had died from anthrax. They fed the vultures, and I can tell you, the vultures in their normal state, everything being equal, the vultures won't die from anthrax because of their makeup, the way the digestive system of vultures. The acid in the stomach of the vultures, because vultures, if you look at their GIT system, is something awesome. They are able to dissolve bones, cartilage, tendons, and they've got the highest acidity. That's why you find that even the fecal matter of vultures sometimes almost pasty white because of the amount of acid which is there. So a study was done by Peter Mandi and the colleagues. They fed vultures with meat with anthrax, just like Kagiso has said. Nothing happened to the vultures. So if the vultures are okay, unless if there is some underlying condition which makes them susceptible, but ordinarily, vultures won't go down with anthrax. It's the same also with hyenas. Yeah.
Oh.
I've seen Karim is happy. I think he knows Peter Mandi very well. Yeah.
Yeah, Prof. Mandi was a phenomenal conservationist and ornithologist. He
Yeah,
was
phenomenal,
actually
absolutely. Yes.
he was my examiner on my master's project, my master's research.
Oh, great.
Yeah, he was a
Ah,
phenomenal man.
wonderful.
Yeah.
Yeah, yeah. Well, a wealth of information and wisdom.
Yes.
There was one time I traveled by road to do some work in Hwange. We sat at Hwange Safaris for 3 days, just tapping into Peter Mandi's knowledge. Yeah, it was fantastic. Yeah, we talked a lot over the anthrax issue. Yeah,
Yeah. I know
yeah.
there's been
It's unfortunate.
a lot of research
Mhm.
recently on the role vultures play in spreading anthrax in the ecosystem.
Spreading anthrax. Yes,
I know for
yes.
for years or for generations we were under the impression that they were quite a big spreader, but I know
Mm,
Campbell Myrne and a few of his colleagues, they did a study
mhm.
to actually show in Kruger that their role was not as or at least has been exaggerated in many cases.
Exaggerated. It's true. I read about that. Yeah, yeah. Absolutely. Yeah.
I think
Wonderful.
Kagiso wants to ask another question.
Yes.
Yes, I
Yeah.
think this was a hand. You can go ahead. Yes.
Yes, Prof. Musso. So I wanted to ask again, so do you think would it be practical if ever we tried to breed more of vultures in our as our management strategy for curbing the spread of anthrax? Would it be a practical idea?
Oh, breeding the vouchers. Ah, that one. I'll ask Karen to help me with to respond to that one because I know the vouchers breeding is not a simple. Eh, go ahead,
So
Karen.
So yes, it is a good thing that vultures don't succumb to things like anthrax. But the reality is,
Yes,
breeding vultures in general in captivity has proven to be very difficult. They don't breed
very
very
difficult.
quickly.
Very
And
difficult. Mm.
I mean the reality now, if we look at all our vultures across the globe, all of them are in a very bad state
They are
because of
reducing
poisoning, because of
bad
poaching,
state.
because of all of that. And
Mm.
so logistically, yeah, unlikely to be a viable option.
Yeah.
Absolutely. Mm.
Okay. And also last year, maybe we can just chat about how they spread the disease. You have also said that they help in spreading it. Of course, they don't suffer, but they help in the spreading. Can you just elaborate so that the student can also get the thing?
Um, like help in spreading the disease or what?
Yeah, I heard you talk with Karen that the vultures, of course, they
Huh.
help clean the environment, but they also help spread the disease. So maybe you can just elaborate on that one.
Oh, yes. Actually, Karen has outlined very well. The concept, it's a bit of a misconception which has been there, is that vouchers are known to have been spreaders. You see, when you're talking about disease, there are so-called normal spreaders, super spreaders, and the vouchers were like exaggerated to almost super spreader status. But like Karen has said, most of the studies have indicated that vouchers actually they are not that bad spreaders of the disease. And these are studies which are being conducted. They are not, their crucial role actually in maintaining the ecosystem is actually higher because these are the cleaning agents of the environment. Without the vouchers, we don't know the amount of disease which would have been in the environment because the vouchers, the role they play is actually under appreciated, but they play a very critical and crucial role in maintaining the balance of the ecosystem. Yes.
Yeah, absolutely. And I think that misconception came
Mhm.
about, especially in terms of anthrax, is because as Prof. Musso mentioned, anthrax, when anthrax is exposed to oxygen, that's when the spores are not only released, but they're increased. And so vultures play a very big role in opening up a lot of your carcasses in the field. And so and when a vulture feeds, they get into the carcass itself. And so the idea was that it would be covered by infected material. And then as soon as that vulture feeds, they immediately go to a water hole, and they'll bathe in the water, they'll drink.
Yes.
And the idea was that this now vulture that's covered in all this infected material will be spreading the spores into the water hole and into the surrounding areas. But again, that's been proven by quite a few studies now to be, if it is happening, it's negligible. The effect is negligible.
Okay.
Absolutely. Absolutely.
Thank you so much. And there is another question from Genesis. She says, is there any indicator species for anthrax?
Yeah, it's
Indicator
very
species,
simple. Yeah, because indicator species, these are species which easily detect the presence of anthrax. And like I've said, these will depend on an area. Like in Zambia, the western part of Zambia, we've got cattle. Cattle themselves are the first ones to show the symptoms that there's anthrax. In the eastern part of Zambia, we've got the Luango and the other herbivores. These can be categorized as indicator species because they will be able to detect through them being affected that there is presence of anthrax. Then they become indicator species. So depending on an area, in some areas it can be maybe some deer, it can be even some rodents. So those animals which show early detection that anthrax has broken out, they become the indicator species. Because indicator species can be at a micro level, up to small mammal, through small mammal to large mammal communities. So it will go from all that. But I will look at the large mammal communities being cattle and the hippos for us in Zambia. Yeah.
Okay. Thank you so much.
Yes.
And there is also another question from Selo. Selo, would you please unmute and ask the question? He says,
he said his network is bad.
Yeah, but the question is, have you come across cases where anthrax is beneficial to the environment or in any way?
I like that one. You say that's a very good one, political question at the same time. When you are talking with a conservationist, they will tell you that it's difficult to say that anthrax is beneficial to the environment, especially looking at the disaster which it is causing, because we look at the loss of life. And I don't know how we can categorize the loss of life being beneficial to the environment. And something which becomes also it's a weapon. You know, anthrax can be weaponized. So all those aspects, we are looking at it as being a danger, actually. It's more of a risk because it's a hazard, in short. So being a hazard to both humans, even to the environment itself, and to the animals, I don't see it being of beneficial value, but it's being more of a biological hazard. Yeah, that's what I can. I've liked this question, but that's my answer. Yeah, we are dealing with a hazard here. Yes.
Thank you so much. Maybe we can also say it helps to maintain the populations, maybe. Because
yeah, what you have said, I didn't want to bring that because I didn't want to come to that. The Luangwa River, there are points where it has the highest hippo population. And the only natural control has been anthrax. You might have heard of that. I did want to bring that one, but anthrax has been controlling the population of the hippos. Karen knows very well about that. Yeah.
Okay. On the same, like, there was a time where I was teaching primary kids about conservation, and we said the biggest threat to the ecosystem is the growing population of people. And this child, during the post survey, she wrote like, we asked, there is a question where you say, do you have any question? And she goes like, what can we do about this population? Maybe should we introduce diseases so that other people can die?
It's difficult. I know it's a difficult situation. Yeah.
It's a very difficult one. Maybe we need an
Absolutely.
answer. Okay, there is another question from Kagiso. Kagiso, are you there?
Yes, Kagiso.
Yes, I am here.
Is muted. Yeah.
I wanted to ask again if has there ever been a case recorded of anthrax in snakes, maybe?
Sarah, it was a bit muffled. Are you able to
Yes, I can
explain
read
what
it.
Kagiso asked? I couldn't hear him clearly. Yes.
Okay, it's
Read
asking
it.
if there have been a case of. Prof, are you there?
I think my network was jumping. Yeah, I'm here. Go ahead.
Okay. Yeah, he's
My
asking if
network.
there have been
Yes.
a case of anthrax in snakes.
Anthrax in what?
Snakes.
So
Snakes.
my network is. Snakes, reptiles.
Yes.
That's a very
Yeah.
good question. That one, I'm going to throw it to my colleague Karen. Maybe he has got information. You see, very few people deal with snakes, and I don't have an answer to that one. I like that question by Kagi, so something we have to look at. Karim, any information on anthrax in snakes?
Yeah, I have never personally heard of any case in reptiles at all. And I think it's not, it's
Absolutely.
more a case of it's difficult to detect it. Snakes, as in general, are quite a cryptic animal to detect, and a sick snake that dies in the environment somewhere is very difficult to retrieve before the carcass deteriorates. And so I think it's more a case of just haven't detected it, but it is definitely something I think worth looking at.
Yeah.
Absolutely. Yeah, because like Karen has said, for snakes, most of the time, once they get their warmth, they are down below the surface. So with these large mammal communities, there is what is also known as optical stimulation, what is going to stimulate your eyes to say, oh, what's happening? But for snake, they are subterranean, they are not there. And it's very difficult. Sometimes we've seen carcasses of snakes, but it's once in a while, and very few people get even stimulated. Sometimes, oh, a snake must have died here. What happened? Then you move on. No research is done. No one gets affected. And that's a very good observation, Kalki. So like Karen has said, it's very difficult. We don't even see this in the real life outside there. Yeah. Thank you, Karen, for that.
Okay. Thank you all. Thank you all for your input. So I was also thinking about this. Yellow mongoose also does get affected by anthrax, right? So I was thinking that maybe for a mongoose to get anthrax, it might have maybe eaten a snake that might have been in contact with anthrax, and that's how maybe we can see the mongoose has anthrax. So am I wrong to think that way or not?
Yeah, that's it's good. Kagiso is a thinker. What do they say? Like a philosopher. He is a philosopher. Yeah, with yellow mongoose, these are very agile animals. And there is something special with the yellow mongoose. I don't have very good information, but I've seen how they respond even to toxins, even to snake bites. And but with the anthrax, I don't have any information. I will ask my colleague, maybe Karen has got something, but so far I've taken note. Thank you, Kagiso. We are now creating more areas for research, but these are very difficult animals to handle, very difficult even to just for you to give you distance to capture them. They are very difficult animals. I don't know, Karen, what you can say about the yellow mongoose.
Yeah, in terms of anthrax, very little information on yellow mongoose. I think anthrax in particular, because of its impact on your larger mammals, how easy it is to pick it up in the larger mammals, we've concentrated a lot on those larger mammals. Not to say that it doesn't occur in the smaller mammals, it's just a lot more difficult to identify it and pick it up. I mean, yellow mongoose in South Africa, in fact, in Southern Africa, in Africa in general, yellow mongoose is one of the major vectors for rabies. So we know that they are susceptible to various different game diseases, and I don't see why they wouldn't be susceptible to anthrax. It just might not be to the same scale as your larger animals.
Yeah, for rabies, yeah, they can be a nuisance, eh? Serious.
Big problem for rabies. Big problems in Gauteng. Even if we narrow it down to the cradle of humankind region in South Africa, we found they're one of the biggest vectors for rabies at the moment.
Yeah, absolutely.
That was a good question, Kagiso.
He's giving us more areas for research. Thank you very much,
Yeah.
Kagiso.
Here we go,
Yeah.
Kagiso. That's a master's and PhD project for you.
Absolutely. I concur. I second that.
Okay, thank you so much. If there is no another question, then this will be the end of our today's presentation. But then there is a comment from our director, Johan, in the chat. I would like to ask him to unmute and talk to us. He should read it out himself.
Yes.
Thank you, Prof. We really, really appreciated this lecture tonight. I would, if you sound like the kind of professor that we would all want if we are sitting in the university benches. So you make me miss my university days. So thank you for this lecture. We really appreciate it.
You are welcome very much, Johan. You are welcome. Thank you very much. I'm in Zambia here. It's dark. We don't have power. I've tried just to make sure that I deliver this lecture. Yeah, we are in darkness here. Thank you very much, Johan, for your good guidings and the encouragements and the support. Thank you very much.
And thank you for the TUT students for the brilliant questions tonight. I mean, that really made the evening come alive. That was fantastic. Really appreciate it. We look forward to doing this again next year.
Absolutely. Thank you very much. Thank you.
Thank you so much. Yeah, thank you, everyone. And thank you, Prof. Today, I'm a very happy person to finally be in your class and be your student after reading all those papers. And thank you so much for your time. You're a busy person, but you make time to come here and be our speaker today. We really appreciate that.